Life in the world's oceans is endangered. In coral reefs, in kelp forests, in marshes and wetlands, in bays and harbors, and along our coastlines. Once abundant marine environments have suffered major damage over the past century from human activities, including overfishing, development, pollution, and runoff. Problems vary worldwide, but the overall diagnosis? The oceans have been injured and may be on the verge of even greater ecological changes. Scientists report that many marine populations and habitats are reaching the point at which even small disturbances can cause big changes. And a threat to one species can have ripple effects that threaten the overall ecosystem. Around the globe, marine scientists are monitoring and evaluating biodiversity, and their findings indicate significant actions must be taken to prevent further loss of ocean life. At Scripps Institution of Oceanography at the University of California, San Diego, researchers in the Center for Marine Biodiversity and Conservation are leaders in efforts to better understand the problems the oceans are facing. Enrique Sala is Deputy Director of the Center and a professor whose research focuses on the impacts of human activities on coastal fisheries.
The coastal oceans are in pretty bad shape in many places of the world because of many different factors that act at the same time. The most important of them is overfishing. Contrary to what people believe, pollution and oil spills are not the worst problems of the coastal oceans, but overfishing is.
Near the southern tip of Baja California is the beautiful town of La Paz, Mexico. Here, Enrique Sala and his colleagues from the Autonomous University of Baja California South are conducting comparative studies of marine communities in the Gulf of California, also known as the Sea of Cortez. The majestic beauty of the Gulf belies the fact that decades of overfishing and other human activities have decimated many species of fish and marine mammals. The Mexican government recognizes its dilemma: do nothing and face the prospect of having a dead sea, or take steps to protect and sustain ocean ecosystems. Today, Enrique and a collecting team head out to mangrove forests on the western side of the Gulf along the southern Baja Peninsula. Co-investigators on the project are Gustavo Paredes, a graduate student at Scripps, and Octavio Alberto of the University in La Paz, who is also now studying at Scripps. They wade out into a lagoon filled with red mangroves. Mangrove plants are salt tolerant, and their long tangled roots provide a perfect habitat for a wide range of marine organisms. The scientists are here to collect juvenile snapper fishes to investigate how successfully they are reproducing in the Gulf. Snappers are hatched as larvae at offshore islands, and as juveniles must make it safely to coastal habitats, such as this one, in order to survive and develop.
This species of snappers, the juvenile phase is found only in these mangroves, and there are very few and very small mangroves in the Baja California side of the Sea of Cortez. So if these small mangroves support the entire population of snappers along the coast of Baja, they are extremely important, more important than we thought before.
This lagoon was once a commercial fish nursery, and today its crumbling walls and waterways that still connect to the lagoon opening serve as protective nursery for a variety of young marine animals that would otherwise be eaten in the open ocean. The walls provide a safe haven for two of the major snapper species in the Gulf, yellow and dog snappers. The snapper larvae become juvenile fish at about a month old and must find a protective coastal habitat such as this in which to grow to near adult size. Working with a seining net, the team closes off a section of the wall and then uses a small dip net to collect the individual fish. Back on the boat, they examine a dog snapper, a species that is rare compared to other snappers. Finding this juvenile is a good sign that some dog snappers are having a productive spawning. Next, they head out to another estuary in a secluded area of the southern Gulf. This area is a network of streams running through a thick mangrove marsh.
This is one of the small mangroves in Espiritu Santo Island. Small stream, very shallow, and it's very difficult to collect the juvenile snappers here because they hide between the roots of the mangroves. But this is a very good habitat, and this habitat you see here is full of juvenile dog and yellow snapper.
Another way they collect the small fish is with a homemade spear gun constructed with a rubber band and bicycle spokes. They spread out along the bank and slowly snorkel into the undergrowth. The shallow intertidal area is a habitat for a high diversity of crabs, shrimps, mollusks, and smaller organisms upon which the juvenile snappers feed. Several yellow snappers have been collected from this site. They're not all the same size because larvae are continuously arriving at the mangroves. The larger snapper is about 2 months old, the smaller about 1 month. As the day stretches on, the team heads out to yet another mangrove forest. The next day at the university, Gustavo prepares to take a closer look at the specimens for analysis of their growth rates. Each fish is measured and labeled with its size, date caught, and other information. Gustavo needs to know the exact age of the fish, so he prepares it for dissection. Using a glass knife that he made by hand, he performs the surgery under a microscope. He is removing a small bone from the base of the fish's ear called an otolith. Flat and round, the otolith forms layers like tree rings. During the juvenile stage, it grows 1 layer a day. Small amounts of the bone will be removed for chemical analysis, and the rest of it is permanently affixed to a glass slide. The mounted slides are a valuable database of information on the nursery of juvenile snappers in Baja's mangroves. These analyses are part of a larger ongoing research effort at the University in La Paz in cooperation with Scripps and the World Wildlife Fund to design a network of marine reserves throughout the Gulf. The reserves would protect spawning and nursery areas from overfishing while supporting the fishing industry by ensuring healthy future fish stocks.
The overall goal of our project in the Gulf of California is to provide the best science to see the implementation of a network of marine protected areas that will protect and preserve the biodiversity of the Sea of Cortez for the long term.
Worldwide, tropical reefs are among the most threatened marine environments. In the Atlantic Ocean, once vibrant Caribbean corals are dying as diseases spread over wider geographical ranges. The die-off is extensive for major species of reef-building corals, and in decimated areas, sediments and seaweeds replace them. Off the Atlantic coast of Panama, near its border with Costa Rica, are the islands of Bocas del Toro. These lush green tropical islands are in an isolated area of Panama and have some of the most unspoiled coral reefs in the Caribbean. Only a few thousand people live in Bocas Town and surrounding villages, but during the past decade, international travelers have discovered the area, resulting in increased population and development that threatens the environment. The Panamanian government and the residents of Bocas are proactive about preserving their paradise, so they are working with concerned scientists from around the world. Among the researchers here is a graduate student from Scripps' Center for Marine Biodiversity and Conservation. Davy Klein is studying how changing environmental conditions are affecting coral reef habitats and what the future may hold.
The reefs are in an incredibly sad state. In the next 20-30 years, if things continue as they're presently going, it's possible that there will be no more healthy Caribbean reefs at all. and I'm trying to figure out how all the things that we're putting into the ocean, the sedimentation associated with cutting down rainforests, the pesticides from farming, the sewage that dumps out into the ocean, the oil from boats, how each of those things affects the coral animal, and more specifically, how it affects the relationship between the coral and the symbionts that live within the coral.
Davy Klein has been living in Bocas del Toro for over a year, working at the Smithsonian's Tropical Research Institute. The Smithsonian has several research stations in Panama, and for 50 years has worked to protect the country's natural resources. Its labs, facilities, and housing are available to scientists from around the world. The dock at Bocas del Toro gives quick access to the local waters. Neelan Kuntz, a Smithsonian research associate, assists with the project. They are setting up for a collection dive off the main island at Punta Caracol, a diverse reef in 15 to 20 feet of water. Davy enters to snorkel around on the surface and pinpoint a good collecting area. They are seeking Monastria annularis, or mountain star coral. It's one of the most common reef-building corals in the Caribbean and the Gulf of Mexico. Each coral colony is made up of thousands of tiny individual animals called polyps that are basically a mouth surrounded by tentacles. Polyps produce a covering of thick mucus that is constantly shed from the coral. The mucus supports a diverse community of bacteria that differs between species of corals. This mucus also serves as the coral's first line of defense against disease. Davy places a syringe on individual polyps to suck off mucus to take back to the lab. Notes are taken about the health of the coral, and the mucus samples are marked and numbered. As their experiment requires living coral, Davy looks over the colony for a healthy sample of the right size. He selects a piece where he can chisel on dead skeleton to avoid damaging any living tissue. The coral colony needs to be flat on the bottom, so it is trimmed with bone clippers, again, just cutting the dead skeleton. Back on the boat, Davy and Neelan examine the samples to check their suitability. About 40 polyps are on this small piece. Despite the extensive damage caused by coral disease, very little is known about what causes them. Of the 27 reported coral diseases, the causes are known for only a few. They head back to the Smithsonian's dock. Here, the corals are placed into a one-of-a-kind aquarium that accommodates 400 samples simultaneously. Designed by Davy and Forrest Rower of San Diego State University, the system exposes the corals to precisely controlled stresses while mimicking the conditions of the reef. Filtered reef water is pumped into the system and mixed with various concentrations of nutrients, chemicals, and bacteria. They're taken from sewage, asphalt, and other pollution sources.
This concentrated chemical is then mixed with the seawater, and the dosed seawater travels down these lines to what we call an accumulator. And what the accumulator does is then distribute the water to the 10 replicate corals, and then we can look at the effects of the chemical on these corals. We can look at 40 independent chemicals on 400 corals, and we can get an idea of what levels of the different stressors affect the corals and how it changes the relationship between the coral and their bacterial and algal symbionts.
A healthy coral colony has a rich brown coloring from its symbiotic algae, while a diseased one is bleached white. In the Smithsonian's laboratory, the mucus samples are prepared. The bacteria in the mucus are introduced into a specific growth medium in petri dishes. Sterile techniques are used to ensure no contamination. The experiments have shown that very low levels of chemicals and nutrients can stress the corals and their symbionts. The dishes are placed into an incubator where the warm temperature will allow the bacteria to flourish over the next 3 days. When they are removed, the dishes contain a thick soup of bacteria, which is transferred into vials of seawater. Neelan then measures the exact bacteria concentration so that they can control the doses added to the corals in the experiment. Davy prepares a sample of asphalt from a local road. Small chunks are broken off and placed into a porous bag. The oils and tars in asphalt and roofing materials dissolve in water, and their runoff is one of the major problems of urbanization near coral reefs. Back at the experimental aquarium, Nealon mixes a bacterial solution into the system. The research has shown that some coral diseases are caused by a loss of balance among bacteria species living on the reef. Davey takes a packet of asphalt and puts it into another container. They've found that asphalt kills corals quickly and are trying to determine which compounds are the least toxic, so recommendations can be made about future road and building materials. The greatest concern is that people who live near coral reefs often don't realize how their actions affect the marine environment.
From my research, it's pretty clear that most of the stuff we're dumping into the water is going to affect the reefs. And if people don't think about what they do with their sewage water, what they do with the runoffs from banana plantations and from farms, then we're going to lose all this diversity associated with reefs.
The future of Caribbean reefs is also closely tied to the ability of corals to successfully reproduce, while ecological conditions continue to change and degrade. Only in the past decade have scientists examined the details of coral reproduction. And in another study at the Smithsonian Tropical Research Institute in Bocas del Toro, Nancy Knowlton, a Scripps professor and director of the Center for Marine Biodiversity and Conservation, investigates how corals propagate in relation to environmental conditions.
Well, when we first started this work, we were trying to find out how many species of corals were really out there, and one of the best ways of distinguishing different species of corals is to see if they have different patterns of reproduction. So our initial studies had to do with determining whether in fact there were barriers to fertilization between the three species that are in this closely related complex of corals. Now we've moved on to looking at fertilization biology in a more general sense, trying to see when successful fertilization takes place and when reproduction is unsuccessful. And that's a big part of what we're doing right now. So every year we come out here and monitor the percentage of eggs that get fertilized under different circumstances.
Joining Nancy Knowlton in her research is Professor Don Leviton, a biologist from Florida State University, who is an expert on marine fertilization. They've been working together in the Caribbean for about a decade. Each year, they come to Panama with a group of students to monitor an amazing event, the mass spawning of corals. During these events, they collect data on the timing of the spawning, the rates of fertilization, and the distances traveled by the eggs. They also collect samples for genetic analysis. Nancy explains the procedures that will be performed while diving on the reefs. In the late afternoon, the research dive team assembles on the station's dock, where they prepare for the evening's work. Living samples of Monastrea annularis, our mountain star coral, are placed into buckets so that they can be taken onto the boats for experiments later in the evening. Once all the preparations are made, the team heads out to the dive site. The corals only spawn 1 or 2 nights each year in the late summer, when they release their eggs and sperm into the water in what is called a mass spawning. The team arrives at a site over a patch of healthy mountain star corals. In the hour before dark, they prepare for the dives. Green glow sticks are activated so that they can be used to light a pathway along the reef where star coral heads are located. The divers will follow this underwater survey line when it is dark. Past experience has revealed that one species begins spawning at 8 and the other 2 at about 10. So there will be 2 periods of intense diving. At about 7:30 p.m., the divers enter the water for the first spawning. 8 members of the team will dive, while 2 others operate a chase boat. The water is warm, but the divers wear full suits because of the long time they will be underwater. Normally, it's pitch dark underwater, and the divers work along the reef with just their flashlights. But tonight, the camera lights make it artificially bright. Large heads of mountain star coral line the reef. The water is somewhat murky because many types of plankton and small animals are active on the reef at night. The divers examine the condition of the polyps. When a coral is ready to reproduce, its surface becomes covered with small pink balls that contain bundles of eggs and sperm. When a colony is ready to release its bundles, a diver marks the coral with a lead-weighted glow stick with an identification number. The water becomes even murkier as hordes of small worms and crustaceans come out to feed on the nutrients in the water. Here you see the release. Those are not individual eggs, but instead are bundles of about 80 to 100 eggs glued together with sperm. Each polyp produces an egg-sperm bundle that is released into the water. These corals cannot self-fertilize, so the bundles float to the surface to mix with other bundles from other colonies. When the coral spawning is at full peak, a diver breaks the glow stick loose so that it can be used to follow a bundle to the surface. The bundles rise at a rate of about a foot a minute, which takes about 15 minutes at this site. You can see the surface is covered with floating egg-sperm bundles. The diver releases the glow stick and returns to the bottom to monitor another coral. At the surface, the glow stick is allowed to float around for about 15 minutes and is then scooped up into the chase boat. A plankton sample is taken to collect the sperm and eggs associated with the coral that the glow stick marks. They mix part of the sample with the eggs from the corals that were brought from the lab to see if the sperm in the water is capable of fertilizing them. They record the glow stick's number and take a GPS position to mark the site. The studies have shown that only those corals that reproduce near the same time as their neighbors are successful in fertilizing. Nancy and the other divers are excited about what they saw.
We got repeat spawning of things that we observed spawning last year, which means we can tell if there's year-to-year consistency in the time that individual colonies spawn. And we also got spawning records for one of the 3 species that previously we've never gotten from this part of Panama, so that was great.
The team goes on to conduct a second round of dives later in the night. At about midnight, they return from the field. It's late, but their work this night is far from over. All of the samples are brought back to the lab for detailed analysis. Individual eggs can easily be seen in the cup. They are placed into a small maze that spreads them out into straight lines. The researchers can tell whether the egg is fertile because after about 3 hours, it is divided into multiple cells. A microscope is used to examine the slides and to score the samples for rates of fertilization. Here you can see some cells that have successfully divided. A counter is used in one hand to score fertilized eggs, while a counter in the other hand logs unsuccessful eggs. Tests are also made on the impacts of various salinity levels on fertilization, as well as the effects of several other environmental conditions. They found this year was an exceptional spawning with success rates of 80, even 90%. The kind of year corals depend on in order to propagate themselves from one generation to another. Yet Nancy Knowlton has great reservations about the coral's long-term future.
A recent study was published showing that about an average of 80% decline in the coverage by corals in the last 3 decades, which is really a very serious level of decline when you consider the fact that adult corals are typically 1 or more centuries old. So these mortality events are not something that reefs can recover from in the course of just a few years or even a few decades.
These 3 studies of the interactions among marine ecosystems and the effects of overfishing in the Gulf of California, of coral diseases and human activities in Panama, and of the population dynamics of Caribbean reefs, are representative of how scientists investigate marine biodiversity. The loss of biological diversity in the oceans is far less publicized than the loss of diversity on land. We've set aside nearly 5% of the United States as wilderness land reserves, whereas only a small fraction of 1% of our ocean waters are protected. Yet biodiversity is an absolute necessity to healthy marine ecosystems and the prevention of both local and regional extinction of many species.
Coral reefs are the most diverse marine ecosystem on the planet. They're comparable to rainforests. They're basically the rainforests of the sea. So in a biodiversity sense, the problem is we're going to lose literally millions of species if we lose coral reefs.
There's also potential cures for diseases found in the corals and the other animals that live on reefs. There's a beauty, and just for future generations, just the experience of swimming on a healthy reef is something that someone remembers for the rest of their life.
All the people enjoying the Gulf of California, be it through recreation or through exploitation, such as fisheries, depend on the Gulf of California. And if the biodiversity in the Gulf of California doesn't have a bright future, these people and their economic activities will not have a bright future either.
In the long term, immediately start planning to reduce the impact of human activities with respect to global climate change, because global climate change in the final analysis could be the worst of all possible threats for coral reefs. In the shorter term, we can do two things. We can reduce overfishing, set up marine reserves where fishing does not take place, and also be very careful about land use so that we don't get lots of sediment running off the land during rainfall and smothering the reef. So deforestation, reducing deforestation, reducing overfishing, and having sustainable fisheries, including marine protected areas, are probably the most important short-term things we can do.
Science is very important in the recovery of biodiversity, but science alone will not solve the problems of the coastal oceans. We need also political will, we need economic incentives, and we need the help of the entire society.
There's no doubt that the oceans were much more bountiful in the decades and centuries that preceded us. We must ask ourselves, what do we want for the oceans of the future? A recent report by the Pew Oceans Commission calls for a new ocean ethic, one in which we become stewards of the oceans and their inhabitants. The Commission's recommendations are that we reorganize and reform the way we govern and manage our oceans, restore our once plentiful ocean fisheries, protect our coastal lands, clean our coastal waters, and establish new aquaculture opportunities. Charting a new course for the oceans won't be easy. It will require a commitment from government, the private sector, and citizens alike. The survival and protection of ocean resources depend on wise economic and political decisions based on scientific knowledge and conservation practices. Scripps Institution of Oceanography is leading the way in scientific research essential for understanding and conserving marine biodiversity. As its scientists discover and learn more, they will find better ways to address the threats faced by our endangered oceans.